So let's talk about synapses and neurotransmitters. When we think of a synapse is going to typically be, in most cases, a physical space in between two adjacent cells that we need to send an action potential across. There are two kinds of synapses in the body. We have chemical and electrical synapses. We're going to spend the vast majority of time talking about chemical synapses because chemical synapses are by far the most common kind of synapse. So as we're looking at an axon of a neuron, I want you to think of it like a wire. And that electric signal, that action potential, will travel down the wire. But if you have a break in your circuit, if you cut the wire, the signal has to stop. We can't send electric signals across gaps in wires. And in our bodies, we have gaps in between most of our neurons. Those gaps are called the synapses. And that when we get to the end of that synapse, or the end of the axon, and we arrive at the synapse, we need to take an electric signal, convert it into a chemical signal. That chemical signal can then travel across the gap, and then when it gets to the next cell will take that chemical signal and turn it into another action potential. So as we're looking at this process, the chemical that we're going to use to send the signal across the gap will transmit a nervous impulse. It is a neurotransmitter. There are many different kinds of neurotransmitters in our body. When we were talking about the muscular system and the neuromuscular junction, we talked about acetylcholine, which is the most common kind of neurotransmitter in our bodies. When we're looking at the two neurons involved with the synapse, some of the terminology we have is the presynaptic neuron and the postsynaptic neuron. The presynaptic neuron is the neuron that comes before the gap. The postsynaptic neuron comes after the gap. And they're going to have the presynaptic membranes and the postsynaptic membranes. This is just some terminology we use to keep our frame of reference. So the signal will always start on the presynaptic neuron, travel to the presynaptic membrane in the synapse, convert to a neurotransmitter. The postsynaptic membrane of the postsynaptic neuron will pick up that neurotransmitter and convert it to another action potential. However, neurons have more than just axons and cell bodies. We also have dendrites. And one of the reasons that the human brain is so powerful is because of all of the connections we can make between neurons. We have multiple dendrites, we have branching of the axons, and these connections can happen across the entire length of our cell. We can have the connections on the dendrites, we can have the connections or the synapses on the cell body, and we can have connections down the length of the axon itself. And because these connections physically aren't that large, we can pack a ton of connections onto a single neuron. So when we think of the neurons, within the spinal cord, we can have up to 10,000 axon terminals interfacing with a single neuron. In other words, we can have 10,000 synapses per neuron in our spinal cord. And then within our brain, where we have lots of processing, it's not uncommon to have 100,000 synapses per neuron. So I know last week we talked about computer processing. We have binary code of a 0 and a 1. And as you alternate zeros and ones, that can be used to process information. Can you imagine how powerful a computer would be if instead of binary code, we had 100,000 transition states for each transistor? You could pack a lot of you get a lot of processing power with a very small number of transistors or processors. And that's what we have within the human body. Right now, if you look at new modern computers, there are more transistors in modern computers than there are neurons in the human brain. But the difference is one neuron can have so many connections that it is capable of performing more processing power than a single transistor within a modern computer. As we look at these synapses, if an axon is bound to a dendrite, we call that an axodendritic synapse. If an axon is bound to the neurosoma, we call that an axosomatic synapse. And if an axon attaches to another axon, we call that an axoaxonic synapse. So if we take the root words associated with the parts of the neuron that can be used to tell us what two parts are connecting together to make that connection, to make the synapse. Now, when we look at the synaptic cleft, this gap, this space that's physically between neurons is a very small space. And it wasn't discovered until the 20th century. And when we're looking at this process, it's worth mentioning that the first instance of neurotransmitters being shown or demonstrated in a laboratory situation was in 1921. So it's still relatively recent that we discovered neurotransmitters. And this is a classic experiment that's been recreated many times in undergraduate and graduate laboratory activities, where you take frogs, living frogs, and then you isolate the tissues from those frogs, and then you perform your experiment on the tissues. If you take, in this case, Otto Loewy took the frog, isolated the hearts while the frogs were still alive, and then put saline around the hearts, and then he used an electrical stimulus to stimulate one of the major nerves of the heart, found that the heart slowed, and then with the second frog, he was able to remove the saline from the frog and was able to recreate the slowing of the heart. And then he named this stuff in honor of the nerve he stimulated, the vagus substance. But however, it was later renamed acetylcholine, the first neurotransmitter. So that's a little history for you on the discovery of acetylcholine. Up until this point, we've talked exclusively about chemical synapses, but any conversation on synapses would be lacking if I didn't mention that we also have electrical synapses. When we think of an electrical synapse, there is no physical space between the cells. One cell membrane will physically connect to another cell membrane with an electrical synapse. And as we're going from one cell, the presynaptic neuron, to the postsynaptic neuron, with this electrical synapse, or the presynaptic cell and the postsynaptic cell, that we need to have the ions continuously flowing from one cell into the next cell so that we can have a continuous wave of depolarization traveling down the cell membranes. And we have a special cell junction that allows this. We talked about gap junctions during Unit 1 when we were talking about cell-to-cell connections, where we talked about gap junctions, tight junctions, and desmosomes. Just to review, the gap junction is kind of like a pore or a hole that connects two adjacent cells. I always think of wiffle balls when I think of gap junctions. And this gap junction will let ions, sodium and potassium ions, flow from the presynaptic cell directly into the cytoplasm of the postsynaptic cell so that we can have a continuous transfer of electrical signal. And there's some advantages and disadvantages of the electrical synapse. The advantage is you get a really quick signal conduction. The signal travels super quickly from one cell to the next cell because fewer things need to happen. With a chemical synapse, you need to let different ions in, you need to exositize neurotransmitters, you need to activate the ion channels in the postsynaptic membrane. There's more steps involved that physically take more time. With electrical synapses, it's just conduction of the charge. And because there's fewer steps involved, we go from one cell to the next cell in a shorter period of time. The big disadvantage, though, is that you have less regulation. If this process of sending the signal is completely automated, we don't have the ability to process the information as quickly, or as much, I should say. So within our brain, we have almost exclusively chemical synapses. And by having chemical synapses in our brain, we can have excitatory neurotransmitters and inhibitory neurotransmitters that can mix and match and allow for the processing of signals, the processing of information at the neurosoma. With an electrical synapse, it's an on or off situation. There is no gradients associated with the electrical synapse, and we can't make any decisions. We can't process the information with electrical synapses. So we typically are going to use them in situations where there is no processing needed. The most common example of an electrical synapse is from one cardiomyocyte to the next cardiomyocyte. once the heart muscle tissue starts contracting, you want all of those muscle cells in one chamber of the heart to contract at the same time. Let's go back to a chemical synapse. So when we're looking at a chemical synapse, our signal, the action potential, travels down the axon. We have sodium voltage-gated channels open, potassium voltage-gated channels open, and it just keeps moving down the axon until we get to the axon terminal of the first neuron, of the presynaptic neuron. And once we get to the axon terminal, we introduce a new voltage-gated channel. We're going to introduce a calcium voltage-gated channel. And at this axon terminal of the presynaptic membrane, or presynaptic neuron, calcium voltage-gated channels open up, calcium rushes in, and then will bind to the synaptic vesicles, or the storage vesicles that are filled with neurotransmitters. When calcium binds to those neurotransmitter, or those storage vesicles, the storage vesicles will exocitize their neurotransmitter into the synaptic cleft, or the space in between the axons. And then as those neurotransmitters are released into the synaptic cleft, they bind to receptors, here the receptors are shown in red, on that postsynaptic membrane. Now as we look at this figure up on the screen is designed to help illustrate a concept. In real life, the synaptic cleft is much smaller, much more intimate. So this figure is overemphasizing the diameter or the distance of the synaptic cleft for the sake of being clearly understood. But it's kind of like the Notaron VA. In real life, it's much, much smaller and very difficult to perceive with your light microscopes. Here's an electron micrograph of a neurosoma with a bunch of axon terminals attached to it. So as we look at these axon terminals, the axon terminals tend to bury themselves or indent themselves into the neurosoma. And this one neurosoma in the background has, from the appearance of this figure, I would guesstimate dozens of axons synapsing with it. So this is a more accurate representation of what happens in real life. This tends to be a little overwhelming when we look at a big picture, so we're going to zoom in and focus on individual synapses of one axon and one neuron. When we look at the chemicals that are sent across the synapse, those chemicals are referred to as neurotransmitters. In our bodies, I can't emphasize it enough, acetylcholine is the most common neurotransmitter. It's the most well-studied, and it even has an entire category. It stands alone. There's a whole category dedicated to just acetylcholine. But as neuroscience has advanced, we've isolated over 100 different chemicals that function as a neurotransmitter. When we think of a neurotransmitter, you can loosely define a neurotransmitter as a chemical that alters the voltage of a membrane. It can either raise the voltage and trigger an action potential, or it can lower the voltage and inhibit the action potential. We have excitatory and inhibitory neurotransmitters in our body that we use to activate or deactivate our action potentials. The major categories of neurotransmitters include acetylcholine, which really just stands by itself as a category of one. Then we have the amino acid base, the monoamine, and then the neuropeptide. There also are some other variations of neurotransmitters or some subcategories, but these are the four big categories. If you look in your lecture textbook, the textbook for this course lists dozens and dozens of different neurotransmitters. Please do not memorize those neurotransmitters. I'll emphasize the key ones I want you to focus on here during lecture. Obviously, acetylcholine has been emphasized, so commit that one to memory. So when we look at acetylcholine is formed from acetic acid and a choline molecule. Here's the molecular structure of the acetylcholine. And as we look at this acetylcholine is broken apart by an enzyme referred to as acetylcholine esterase. It will cleave the acetylcholine at the ester bond right here, leaving acetic acid and a choline molecule left over. When we look at amino acid-based neurotransmitters, these may or may not be the amino acids that we use in protein synthesis. To make proteins in a human being, there are 20 different amino acids that we can mix and match, kind of like different kinds of Lego bricks that we can snap together to make the protein molecule. But when we look at, chemically speaking, there can be potentially an infinite number of amino acids produced. It's anything that has an acidic side chain, a carboxylic acid, side group, along with an amine side group, and then you can mix and match and attach whatever the heck you want onto that middle carbon. So in reality, there is an infinite number of amino acids, but I'm going to emphasize this again. In human proteins, we only use 20 amino acids. Some amino acids that we have in our bodies aren't used in proteins. They're used as neurotransmitters. Other amino acids can be used as both, built-in block of a protein and a neurotransmitter. As we're looking at these, glycine, aspartic acid, and glutamic acid are amino acids that we use as both neurotransmitters and building blocks of proteins. They're dual function amino acids. When we look at GABA, this amino acid, gamma-aminobutyric acid, is only used as a neurotransmitter. When we look at this abbreviation right there, that is the Greek letter gamma for gamma-aminobutyric acid, or GABA. And that's an amino acid that's a neurotransmitter only and not used for protein synthesis. Some other neurotransmitters are going to be the monoamines. And when we look at the root words of this name, mono means one. Amine refers to an organic functional group that contains nitrogen. So when we look at our monoamines, they need to have an NH2, an amino functional group. And then they're also going to have their carboxylic acid functional group removed. Because they've had their carboxylic acid functional group removed, these are not amino acids, they're just monoamines. The most common monoamines in our bodies include epinephrine, which is also known as adrenaline, norepinephrine, also known as noradrenaline, dopamine, and serotonin. So when we look at epinephrine and norepinephrine, I want you to think of the hormones associated with stress. These are the hormones. It looks like I can't write over there. So I meant to write stress up on the screen. Those hormones, the epinephrine and norepinephrine, are associated with the sympathetic nervous system response and are primarily going to be released from the adrenal glands. When we look at dopamine and serotonin, those are neurotransmitters that are primarily going to be active within the brain and help to regulate memory formation and mood. And when we look at histamine, this is a dual-purpose neurotransmitter. Clinically speaking, we spend most of the time focusing on the inflammation response associated with histamine. We also have neuropeptide-based neurotransmitters. These neuropeptide-based neurotransmitters are oligopeptides. They're like small protein molecules. And if you try to find a clear distinction between what makes an oligopeptide and a polypeptide, most textbooks are going to waffle. It's kind of a gray definition. It's not black and white. So we're tentatively just call these oligopeptides because they don't have hundreds of amino acids. They have dozens or less than 30 amino acids typically to make up these neuropeptides. When we look at these neuropeptides, these are going to be neurotransmitters that sometimes function as just a neurotransmitter. Sometimes they function as a hormone. Sometimes they're both a neurotransmitter and a hormone. Two that are worth emphasizing are cholecytokinin. Cholecytokinin is a chemical that is both a neurotransmitter and a hormone, and it's primarily involved with regulating digestive functions. We'll talk about cholecytokinin all the time when we get to the digestive system in Bio 314 next semester. That's a very key component, or key player for regulating digestion. And we're also going to have substance P as well. Substance P is an additional neurotransmitter that helps to regulate digestive functions. We also have gases that are used as neurotransmitters in our body. One of the key things to emphasize of these gas-based neurotransmitters is they have to be synthesized on the point of release. We can't make the gas in advance and store it. Making a gas in our body and storing it in advance is very difficult to do because gases have a tendency to rapidly diffuse across a phospholipid bilayer. So when we make a gas, it tends to dissipate. It's really hard to keep localized. So our body gets around this by synthesizing it at the point that we need it, right when we need it. The two most common gas-based neurotransmitters are carbon monoxide with one oxygen and nitric oxide, sometimes also referred to as nitrogen monoxide, to use the correct current nomenclature. So when we look at carbon monoxide and nitrogen monoxide, these are two gases that can diffuse rapidly throughout the cell and act as a neurotransmitter. It's worth emphasizing, particularly with nitric oxide, that this neurotransmitter primarily will act as an inhibitor of smooth muscle contraction. So clinically speaking, when we think of individuals that have high blood pressure, a lot of times they take nitroglycerin tablets. And they use those nitroglycerin tablets to cause the synthesis of nitrogen monoxide to be upregulated, and then the smooth muscle in their blood vessels relaxes and their blood pressure goes down. I'll talk about Viagra with the reproductive system. It is connected to nitrogen oxide. It's a funny story. I don't want to steal my thunder from next semester. So let's talk about synapses in our bodies. When we look at our synapses, some synapses will have excitatory neurotransmitters that take that membrane voltage and bring it closer to threshold. Some synapses have the inhibitory neurotransmitters that lower the membrane voltage. And when we think of excitatory neurotransmitters, those excitatory neurotransmitters and synapses primarily let sodium into the cell to raise the voltage. When we think of those inhibitory neurotransmitters, they are primarily going to let chlorine ions or chloride ions, a -1 chlorine ion, into the cell. And by letting those -1 chlorines into the cell, you lower the membrane voltage. So we'll talk about the excitatory, the inhibitory, and another excitatory. First, let's focus on the cholinergic synapse. These synapses primarily are going to use acetylcholine. They are the most common synapse in the skeletal system. And to review again, because we already covered this with the skeletal system, when we get to an acetylcholine synapse, when the action potential gets to the end of the axon, calcium ion voltage-gated channels open up, calcium ions rush in, they bind to synaptic vesicles. Those synaptic vesicles will merge with the synapse, presynaptic membrane, and exositize acetylcholine. And then acetylcholine binds to the acetylcholine receptors. The acetylcholine receptor opens up when two acetylcholines have attached to it. And as it opens up, it lets sodium rush in and potassium rush out to trigger the action potential. So yeah, that was everything on this slide. So to refresh, because I went kind of fast, let's cover it again in red. When the action potential gets to the axon terminal of an acetylcholine-based synapse or a cholinergic synapse, calcium voltage-gated channels open up, let calcium in. It binds to the synaptic vesicle, which then exositizes acetylcholine. Acetylcholine attaches to the acetylcholine receptor, or the ligand-gated ion channel. In this case, it's designed to receive acetylcholine. Sodium rushes in, potassium rushes out, and that triggers a wave of depolarization on the postsynaptic neuron, or another action potential. As long as we have acetylcholine in the gap, we're going to be triggering new action potentials because we're going to allow for the flux of sodium and potassium ions, or the movement of sodium and potassium ions across the membrane. So to deactivate these signals, we need an enzyme referred to as acetylcholine esterase. This is a review from the skeletal system. Acetylcholine esterase takes acetylcholine, chops it in half, and then the presynaptic neuron will reabsorb the choline half and then recycle it to make a new acetylcholine molecule. We also have inhibitory synapses, GABAergic synapses. These are synapses that are going to employ the amino acid gamma-aminobutyric acid, also abbreviated as GABA for short. And when we get to this kind of a synapse, the action potential that gets to the axon terminal is going to release GABA, or gamma-aminobutyric acid, into the synapse. And the receptor for this neurotransmitter, when activated, does not let sodium and potassium move across the cell membrane. Instead, this is a protein channel that is designed to let chloride ions travel across the membrane. In particular, chloride ions enter the cell when a GABA receptor is activated. By letting negative ions into the cell, we lower the voltage of the membrane and temporarily induce a hyperpolarized state. We temporarily induce a relative refractory period. It makes it difficult to initiate a new action potential. We inhibit the signal. We also have androgenic synapses. These are synapses that employ adrenaline. Now it's more commonly referred to as epinephrine. And they also are going to respond to norepinephrine as well. So as we look at these synapses, these are going to be synapses that respond to the secondary messenger system. We talked briefly about that, I believe, in Chapter 2 of Unit 1 of this class, way back in February. When we have chemical receptors that can't travel across the cell membrane, they bind to a receptor on the surface of the cell, typically a G-coupled protein cascade. And when the receptor on the surface of the cell receives that neurotransmitter, it changes shape. And on the inside of the cell, it releases the G protein. The G protein will then cause a cascade that amplifies secondary messengers within the cell. During Unit 1, I emphasized calcium ions and cyclic adenosine monophosphate, or CAMP, as secondary messengers inside of the cell. And when we look at the androgenic synapse, cyclic AMP is the secondary messenger of choice, and that cyclic AMP is going to help to amplify the signal. One G protein that's activated can make multiple cyclic AMP molecules inside of the cytoplasm of the cell. Practically speaking, this means that we have an amplification of the signal. A very tiny concentration of these neurotransmitters, the adrenaline and noradrenaline, or epinephrine and norepinephrine, produces a profound effect. It's not uncommon for individuals to feel adrenaline moving through their bloodstream when they get scared or excited or angry. And it's almost like a tingling sensation that travels across their body as the adrenaline is rapidly activating their cells. Now, yes, we have enzyme amplification, so a small signal, a very small amount of epinephrine and norepinephrine gives us a profound effect. But because there are more steps involved with this process, at a cellular level, it takes a little bit longer from the signal to go outside of the cell to inside of the cell because of those protein cascades we need to work through, all of those secondary messengers. Let's talk about turning off a signal. To make a signal stop, the best thing you could do is stop releasing neurotransmitters into the synapse and then remove the neurotransmitters that are already present in the synapse. Neurotransmitters, generally speaking, are going to attach and then detach, attach, detach, and they'll alternate between attached and detached with a period of time of about 1 millisecond being attached to the axon, being attached to its receptor. And then while that neurotransmitter is detached, two things could happen to it. It could diffuse out of the synapse. It can just float away. If the neurotransmitter just floats away or diffuses away from the synapse, it's no longer going to be able to activate an action potential on the postsynaptic membrane. The second thing that could happen is the neurotransmitter could be digested or destroyed by an enzyme present in the synapse designed to get rid of the neurotransmitter. So let's talk about acetylcholinesterase again. I'll go quickly since this is probably, I think, the third time this lecture I've talked about it. So if we look at a cholinergic synapse where we have acetylcholine present in it, to make that signal stop, when the acetylcholine detaches, acetylcholinesterase will bind to it. As acetylcholinesterase binds to acetylcholine, it breaks it into a choline molecule and an acetate molecule. The acetate molecule isn't worth recycling. The carbons and oxygens aren't that metabolically significant. However, the nitrogen present in the choline is kind of hard to get a hold of in our bodies. So we have a tendency to recycle the choline, bring it back into the presynaptic cell, and use that choline to make a new acetylcholine molecule. Those neurotransmitters that acetylcholinesterase does not get rid of are going to diffuse out into the extracellular fluid. Within the central nervous system, the extracellular fluid is called cerebral spinal fluid, and we're going to have one of the neuroglia, particularly the astrocyte, will reabsorb acetylcholine that's just floating around within the cerebral spinal fluid. This is one of those multiple things that the astrocytes will do for us. It helps to maintain the chemical composition of the cerebral spinal fluid. In addition to regulating ion concentrations, astrocytes also help maintain the cerebral spinal fluid by removing excess neurotransmitters that may be floating around within the cerebral spinal fluid. We also have neuromodulators in our bodies. When we think of a neuromodulator, I want you to think of a chemical that instead of having a short-term effect like a neurotransmitter, which is going to be on milliseconds to seconds, a neuromodulator will have a much longer effect. And this is going to be hours to days in terms of the much longer effect. These neuromodulators are going to affect the postsynaptic membrane by altering its sensitivity to other neuromodulators and other neurotransmitters. When we look at nitric oxide, this nitric oxide is one of the neurotransmitters that also doubles as a neuromodulator. It's one of the gas-based neurotransmitters we talked about a couple slides ago. It's going to activate the secondary messenger cascades, and the classic example that's used is relaxing smooth muscle, primarily smooth muscle within the blood vessels. When we look at the neuropeptides that we talked about, they can act as a neurotransmitter by regulating the perception of pain. There are enkephalins and endorphins within our bodies. Most of you probably haven't heard of enkephalins before. However, most of you probably have heard of endorphins. These are chemicals that help to inhibit pain perception. So if you have a high concentration of endorphins being released or enkephalins, your ability to perceive pain is going to be temporarily reduced. Sometimes you've probably heard of the phrase the runner's high, where if somebody's going for a really long race or a really long run, they can't feel the pain, but then the next morning, oh my gosh, when they lose those endorphins, they have a lot of pain associated with that and a lot more regret, I guess you could call it, as they perceive that pain. And that is all we have on neurotransmitters and synapses. Wednesday, we'll pick up with the spinal cord. Have a great day.